to the ladies section again, and this time with Professor Goodeve, and the first time she visits and speaks in the IGMC. So welcome to her. Anne is from Sheffield Diagnostic Genetic Service, Sheffield Children's NHS Foundation Trust, from Sheffield, from United Kingdom. And Anne is going to talk about her experience with the NGS as analysis advanced tool in her lab. The title is Experience of Next Generation DNA Sequencing Analysis in a UK Genetics Laboratory. Down to you, Anne.
Thank you very much. I'll just try and get the slides up. Okay, I'll start by thanking the organisers very much for the opportunity to come and visit Saudi Arabia. It's my first visit, and I'm very happy that I've been able to catch up with some of our previous PhD students from Sheffield while I'm here. So I'm going to talk on an experience from part of our genetics diagnostic service. So I spend half of my time working at Sheffield Diagnostic Genetics Service as a clinical scientist. I'm a member of staff of about 100 within our service, and we undertake both molecular and cytogenetic analysis within the service. And I look after an area that includes bleeding and thrombotic disorders and some of the inherited anemias particularly. So I'm going to talk about a couple of our next generation sequencing panels to you today. So the aims of this piece of work were to introduce next generation sequencing panels for Fanconi anemia and for coagulation and platelets gene disorders. And we started next generation sequencing in our service about 2 years ago. So the first disorders that we introduced were for connective tissue disorders and for inborn errors of metabolism. And over the last couple of years, they've been followed by about 10 other panels for specific types of disorders, with between about 10 and 100 genes in those panels. So, a small bit of introduction just to these two sets of disorders. So the coagulation and platelet bleeding disorders can often be diagnosed through phenotypic testing undertaken by a coagulation laboratory, which means that we often have sufficient information to be able to target one or a small number of genes associated with a particular disorder. Fanconi anemia is the second disorder that I'm going to speak about, and that can result in bone marrow failure. And there is a phenotypic test that can be undertaken, which looks at chromosome fragility testing, and this may be undertaken prior to genetic testing to confirm an individual's phenotype. But in our experience, it's done fairly rarely, so the phenotype often hasn't been pinned down that well when the patients are referred to us. So I'll start with the hemostatic and platelet gene list. So these are mostly bleeding disorders, but not entirely. So Adam TS13 causes a thrombotic disorder, whereas most of these are associated with bleeding, apart from the 3 fibrinogen genes, which can be associated with bleeding or with thrombosis. And in addition to being able to analyse any of these genes independently, we can also group them where we have maybe an unknown disorder. So, for example, for factor 8 and factor 9, if we have a family with a history of haemophilia, but we don't know the type of haemophilia because the male who's affected is long dead, then we can analyse both factor 8 and factor 9 simultaneously. If we have an individual with a reduced factor 8 level, but insufficient family history to be able to say whether it's likely to be X-linked haemophilia or autosomally inherited type 2N von Willebrand disease, we can analyse both those genes together. We can analyse both genes for factor 13 deficiency simultaneously, all of the genes associated with the fibrinogen disorders, and both of those associated with Glanzmann thrombasthenia together. And this has enabled us to speed up our diagnostic process. These are the machines that we use. So we initially started out using the Illumina MySeq, and over the last year have introduced use of the HiSeq. So generally we run the HiSeq machine about once a fortnight, and we try to fill that up with 96 samples where possible to make the best use of the machine. The data analysis then takes a couple of days to reach us. So this is an example of a very simplified example of the workflow that we use. So we follow the gene analysis toolkit designed by the Broad Institute and use a slight modification from our in-house bioinformaticians. So we start with all the variants that were identified on the panel, limit them then to the variants within our genes of interest, and generally with use of this panel we may only be looking up to 3 out of the 13 genes. Any of the frequent variants noted on the sequence variant databases without any pathogenicity attached are removed by the analysis pipeline. So all the top part of this analysis is undertaken through the bioinformatics pipeline, and then the variants identified at the end of that process that may be pathogenic are analysed by our clinical scientists, including myself. We then undertake a pathogenicity assessment that I'll come to in a moment, and end up possibly with a small number of variants. So for haemophilia, probably only 1. For some of the autosomal disorders, possibly 2 variants at the end of that process. So it's one of the simplest pipelines that we can have for our next generation sequencing. So the top example on here shows a patient with haemophilia. So we've got the defect in the factor VIII gene. It gives us the reference sequence, the exon, the nucleotide, and the amino acid substitution. Shows the genomic location for that variant, and the original and the variant's nucleotide. And these further examples just show an individual that we analysed for a fibrinogen disorder, and the variants identified, with the lower one being a common pathogenic variant. So once we've undertaken our next-gen sequencing, we use Sanger sequencing to confirm any potentially pathogenic variants. During the validation of the hemostasis panel, we reanalysed a number of patients that had previously had Sanger sequencing undertaken. We only analysed the genes of interest which had previously been investigated. And our criteria for having full coverage were that we had 30-fold sequence coverage for our regions of interest, which were the exons plus or minus 5 base pairs, and then 18-fold coverage at least for the flanking intronic sequence out to 25 base pairs. And we had complete concordance of the variants previously identified in those individuals, which were between 2 and 30 variants per individual, with the 30 variants being in von Willebrand factor. We were particularly interested in von Willebrand factor as it has a partial pseudogene, so the coding genes on chromosome 12 and the pseudogene on chromosome 22. And it covers a region of about 13 exons of von Willebrand factor in some of the main binding sites for von Willebrand factor. So we made sure that we included 5 patients within the analysis for this region, and these 7 variants shown were identified in the 5 individuals within the pseudogene, within the region that's copied in the pseudogene. And these 2 regions have got a 97% sequence similarity. But we were able to identify the 7 variants previously seen on Sanger sequencing and didn't identify any additional variants within that region in these patients. And subsequently, we've been able to identify 2 gene conversions in this region, which are where part of the pseudogene sequence invades the coding gene and introduces changes into it. So we're very happy that we can uniquely identify the von Willebrand factor coding gene without interference from its pseudogene. For our pathogenicity analysis, we follow the recommendations of the UK Clinical Genetic Science Association. We document all our variants using a so-called unclassified variant form. We start with our local-specific databases to see whether any variants have been identified in our gene of interest, and also use the human gene mutation database. We look at variants identified on the exact exome sequencing project and the single nucleotide polymorphism database as well. If necessary, we undertake in silico predictions using the software Alamut, which provides links to a number of different software tools. And we also have an in-house tool referred to as SNPbot, which has been designed by one of our bioinformatics trainees, and it links to several of the online tools to help with pathogenicity predictions, particularly for amino acid substitutions. And the link at the bottom is to the recommendations that we use. So the patients analysed to date for the haemostasis panel are showed on this slide, and the ones which we look at particularly commonly are those for the haemophilias and for von Willebrand factor. So they're our main genes that we analyse on a more regular basis. And you'll see for a number of the areas that the pickup rates are good for mutations in these disorders because of the phenotypic analysis that's already been undertaken. Our overall detection rate for the mutations in these individuals is 70%. So our impact on the bleeding disorder testing is that von Willebrand factor, which has 52 exons, and we previously undertook by Sanger sequencing as 55 separate amplicons, is now much more straightforward. And rather than analysing specific regions of the gene associated with particular phenotypes, we just look at the entire gene in virtually all of the patients referred. We can look at carriers of unknown haemophilia types where we haven't got an effective male available in the family. And we can look at disorders such as 2N von Willebrand disease and mild haemophilia A together, rather than having to look at the genes sequentially. And where there's more than one gene associated with the disorder, we can just analyse the lot together. I didn't mention, but on the previous slide, one of the individuals that we were able to look at was a baby that had died shortly after birth of a bleeding disorder. And we were asked to look at a number of different possible causes for that, and we're able to just look at all the 5 genes that they requested at the same time, speeding up the analysis. So very briefly on Fanconi anemia. This is an inherited disorder, generally autosomal recessively inherited in all but one gene. It has features of short stature, cafe au lait spots, often thumbs that are poorly attached and non-functional, and other limb deformities in several of the affected individuals. And as well as its features that I've just mentioned, often abnormal gonads as well, and microcephaly, so a small head. So in Fanconi anemia, it can lead to bone marrow failure and cancer susceptibility, and patients are often referred to us for genetic analysis to look at the cause of the bone marrow failure in that individual, and is it due to Fanconi anemia. So we can attempt to identify the causative mutation in the index case, look at relatives subsequently where we can identify a mutation to see whether they're relevant as a bone marrow donor, and also look at relatives to see whether they have an elevated cancer risk because of some of the genes that are associated with the disorder. This is is the current list of the 16 genes that we analyse at the moment, and a number of these you may recognise as being associated with a cancer predisposition, such as the BRCA2 gene. This next slide summarises our findings to date using the next generation sequencing. So the majority of the mutations that we've identified are in the FANK-A gene, which has a predominance of mutations for the disorder. But we've also identified mutations in a further 7 of the genes. The majority are homozygous or compound heterozygous individuals. One individual had 3 potentially disease-causing mutations, and a proportion are just heterozygous for their substitutions. So in these disorders we can have reversion, so sometimes you may have had a pathogenic mutation that's then been lost. So that may be a feature of some of these individuals, but it's difficult to tell when you only find a heterozygous change. And of this group of individuals that we've looked at, amongst 31 patients, we've identified 10 of those to have Fanconi anemia, giving us a pick-up rate of 32% amongst these individuals. To continue our work in this area, we've designed and are just starting to validate an extended bone marrow failure syndrome panel. So we now have further Fanconi genes identified. There's 3 newer ones that have been identified recently. And we're adding genes for severe congenital neutropenia, dyskeratosis congenita, diamond black family anemia, and congenital thrombocytopenia as well to our analysis. And we hope to launch that February or March time next year, once we've finished all of the validation work on it. So in summary, next generation DNA sequencing provides a single workflow for analysis for multiple gene panels. The simultaneous analysis of all the genes associated with the disorder enables speeding up of the diagnostic process. The data analysis is straightforward once the common sequence variants have been removed, and the analysis costs are often reduced. And lastly, I'd like to thank many of the staff in our service that were involved in the next generation sequencing, particularly Kevin Bly and Lucy Crooks, our bioinformaticians who've been involved in setting up the next generation sequencing data analysis pipeline, but also the many others involved in this process. Thank you very much.
Thank you very much, and it's a pleasure to have you here. We have one question, if you don't mind.
Thank you for the presentation. I just wonder if the old tests that we used to use for Fanconi, the mitomycin C and the EB test, the detection rate of those tests is probably much higher than the panels. You say the panels only detect 50% of the cases.
Sorry, could you speak a bit more slowly? I'm missing the last bit of what you said.
Yeah, regarding the mitomycin C and the EB test, the detection rate probably is much higher than these panels. I don't have a clear figure how much is, but for sure it's more than 50%. You say the detection rate using these 19 genes for Fanconi is only 50%. So my question is really clinically indicated to use these panels having a better test before? And my second question is...
Can we just do the first one?
No, and this is very related. The exome sequencing wouldn't detect more than these panels?
So with the phenotypic testing first for the chromosome breakage, often when samples are referred to us, they haven't had that testing undertaken before the samples are sent to us. So if that phenotypic testing hasn't been undertaken, there may be a different disorder than Fanconi anemia present in the patient.
Okay, that's fine.
Does that answer your question?
Yeah, I guess so. If there are no Fanconi, of course the detection rate will be less.
Okay, and The second part for whole exome sequencing?
Yeah, it will be the same. I mean, you don't know if they are in Fanconi, you are doing exome, probably the detection rate will be low. I mean, no, 50%.
So, as part of our service, we don't currently offer whole exome or whole genome sequencing, although we do undertake those as research services, but we haven't moved into undertaking that as part of our own service. Yes, if they didn't have one of the Fanconi genes as the cause for their disorder, it's quite probable that you would pick that up through using a larger panel for sequencing.
Okay, thank you very much. Any more questions, either from the men's side or ladies' side? If not, then we have you.
Yes, hello, I need a question.
Yes, go on.
First, I want to thank you for the very nice presentation. Second, you mentioned here that there are some patients which you were not able to diagnose by molecular.
Sorry, not able to diagnose
by the molecular technique you're doing here.
Yes, that's right.
And if are the patients who are heterozygous, and they are in the clinical picture is typical for Fanconi. In these cases, what do you do?
The only other analysis we have available currently is dosage analysis. So we can currently undertake dosage analysis just for the Fanconi A gene. The main project that our bioinformatics team are working on at the moment is to be able to introduce copy number variation so that from the next generation sequencing outputs we can get not only sequence analysis data but also copy number variation data so that we're able to look for large deletions and duplications as well. So we're hoping that that will be available early next year, but it's difficult to offer that currently.
Thank you.
Any more questions? Right. We have few words from